The first lecture will really be a lecture. We try not to have too many modules which don't have a lot of exercises or coding in them, but this one really is going to be an introduction. Networks, biochemical and other networks are a big subject. You could give easily a whole workshop on them, and in fact our collaborator and friend Herbert Sauro does that every year. He teaches a week-long workshop on this kind of modeling, and we're going to be doing in one day a brief summary of the kinds of things that Herbert teaches over the course of a week. As always, I need to remind you that this module is live streamed and recorded and available for future viewing on YouTube. There are no demos today, but if you're interested in getting ready for the rest of the day, there are cheat sheets, demos, and everything else available in the student materials folder. And I do recommend Herbert Sauro's textbook, Systems Biology and Introduction to Pathway Modeling, as a good reference. So today, also, I should thank our funding sources, NIH and NSF. Today, I'd like to talk a little bit about networks. We're going to start out by just talking about networks in general, and then we are going to begin to use the antimony network model specification language inside of CompuCell and gradually build up to using that more fluently by the end of the day. Today, to begin with, we're going to think about the kinds of networks we might be able to model. We'll look a little bit more detail into some of the kinds of questions you could ask and the mathematics of a variety of network models types. And then we are going to move forward in the remaining modules with understanding how to do those network representations computably. Networks are, of course, one of the great results of modern omics technologies, but converting those network diagrams that you see so often into computable form isn't so trivial. We're not going to be talking today about the software that exists to try to help you do that. There are many, many packages available that will claim to be able to reconstruct networks automatically from molecular data and to try to represent those networks in computable form. They are often helpful, but there is unfortunately no magic. One still has to do a lot of work by hand, and there's still a lot of judgment associated with that. Today, we're going to be focusing on very simple aspects of networks. We're going to be using the antimony model specification language. There's a very nice online tutorial for this and manual. If you go to, it's rather a long thing to type, but if you Google tellurium and then networks, you'll find immediately the web page. I'm not a big fan of naming software commonly used words like tellurium, the element, because it makes things sometimes a little bit harder to find. But in this case, you can find it. There also is a very simple cheat sheet in the student materials folder that you could look at. This is software that was developed primarily by Herbert Sauro at University of Washington. He has a nice introductory textbook called Systems Biology and Introduction to Pathway Modeling. There also are other textbooks of his that are very nice. And of course, there are dozens of textbooks these days on network modeling of various kinds. So when we come to thinking about the kinds of networks that we might want to model in the context of multicellular modeling, there are several. The three that we run into inside of cells classically would be chemical reaction and metabolic networks. Those create and destroy chemical compounds, build the components of cells. They typically operate on very short time scales. Then there are signaling networks that particularly convey information from the surface of the cell into the cell through receptors. And they operate especially with kinases and phosphatases as components. Those operate on slightly longer time scales, seconds to minutes. We have what are called gene regulatory networks, or more generally regulatory networks, that turn genes on and off. There are a whole set of background processes that turn genes on and off that are not that well understood, like acetylation or methylation of histones, methylation of DNA to turn DNA regions off, reorganization of the architecture of the nucleus, which have much longer term effects on turning genes on and off, and which typically are not modeled, although they may be very important. These days, we know, for example, small interfering RNAs are very important to turning the production of proteins on and off. So post-transcriptional control. That again tends not to be modeled so much because it's really not understood so well. So we tend to stick with classic models of gene regulation. Because genes take time to be transcribed and translated, the time scale for gene regulatory networks tends to be from tens of minutes to hours, potentially very long time if we're talking about actual things like acetylation or methylation of histones or DNA. A whole other kind of network that we won't address so much in this class, but that's one of the most common kinds of networks that people use in the pharmaceutical industry when they do mathematical modeling, is what are called physiologically-based pharmacokinetic models. Those model the transport, metabolism, elimination, and absorption, so-called ADME, of molecules in the body. If you take a pill and you want to know how long does it take for the molecules in that pill to get to a particular organ, you want to know how much of the particular molecule gets to a particular region of the body. If you want to evaluate how effective it's going to be or what kinds of side effects it's going to be, those are the models that are used. There are a bunch of commercial companies that make software to do this. The actual modeling itself is fairly straightforward. The art in those models is knowing what the constants are, what the parameters are in those kinds of models. And those are models that all could be implemented, if you know what they are, inside of CompuCell. And so we're really not going to talk, except this morning, a little bit about how to build models of that kind. We're going to assume that you have some idea of what those are. I'm going to focus today on how to implement them once you know what they are. There are other more abstract models, perhaps. They're population dynamics models, which describe the behavior, for example, of viral replication, or for that matter of cells, if we didn't want to do the kind of spatial modeling that we do. Here's an idea of a spatial model of the colonic crypt. We loaded a colonic crypt model last week as a demo, but one could build a non-spatial model where you simply say that each colonic crypt has some stem cells that replicate. Those stem cells become differentiated, partially differentiated cells, which also replicate. Those partially differentiated cells differentiate and eventually die. And so those kind of models are very common. They're much simpler than the models we build in this course. They run a lot faster, and therefore they can be quite useful in a variety of contexts. Another concept that is used, especially in toxicology, but which we'll run into inevitably every time we use a network model inside of the context of CompuCell, is what's called an outcome network. In concepts of toxicology, it's called an adverse outcome pathway. And that could be a series of steps which have a particular consequence, which don't necessarily tie to a particular molecule. So we know that the concentration of a growth factor is correlated with the rate of cell cycle, cell proliferation. But we may not represent all of the connections between a particular molecular species and its behavioral outcome. This is pretty common. You have a toxicant, you say that the probability of a cell dying goes up as the level of the toxicant goes up, but we don't actually model all of the steps involving, for example, p53 or caspases that are involved in cell death. Inevitably, we leave things out. And so outcome pathways are an attempt to formalize the steps in these kinds of sequential processes to be able to quantify them. And we definitely will use those even without thinking about it, perhaps, in the models that we build here. In fact, one of the biggest problems that we have, and I mentioned this last week also, is that the wealth of molecular data that modern omics gives us is not easy to connect with the outcomes that we care about. And we know technically, in sort of a mathematical theorem sense, I can have two cells that have identical molecular composition if I were going to put them through mass spectrometry, but that are completely different in their behavior because those molecules are organized differently. And so molecular concentrations, which are easy to measure, may or may not deterministically or in a simple way tell us what the behavioral control of the cell is. And this is something that we have to live with. This is scientific uncertainty that we have to live with. Again, these networks operate together. We have to remember that all of these networks are always operating inside of a cell. And there's, again, a whole set of spatial organization, which isn't even thought about when you write networks, qua networks. But we do have to remember also that we're trying to build minimal models. And so we very often neglect, for example, most of the metabolic models. Unless we're interested very specifically in metabolic disruption, we tend to assume, and I used this metaphor last week, if you're driving your car, you don't really want to think about how the spark plugs or the fuel injection are working in the car when you're driving. When you push, you put your foot on the accelerator, or put your foot on the brake, what you care about is how the car accelerates, how it stops, whether it stalls, whether it skids. Those things don't depend on the particular details of the operation of the engine. On the other hand, if the engine weren't operating, the car wouldn't work. And so metabolism tends to be something that we take for granted as an engine that works in cells until it doesn't. And we have to ask questions about it. Much of the signaling transport mechanisms within the cell, we take for granted for the most part, unless they're perturbed in the particular problem we care about. And so one of the things in model design that's going to be very important is trying to eliminate the things that are not going to change or going to be critical during the simulation that we're interested in. There is also an issue of timescales, that the fastest process that we model in a system determines the time step of the system. And the faster that time step is, the more expensive the computer simulation is going to be. And so if we have to model electron transport, for example, in quinones in the mitochondria, which happens on a time scale of nanoseconds to microseconds, then we're certainly not going to be able to model what happens over time scales of years. And this is a problem we face, for example, if we ever try to do molecular dynamics simulations, where we can do beautiful, detailed all-atom simulations of molecules and their interactions. But we can only simulate very, very short timescales for large simulations. There's no way to simulate timescales that are meaningful for protein signaling or for gene regulation. It's easy to put up a diagram of metabolic networks. There used to be a wall chart that was produced by a company that sold molecular biology products. And as time has gone on, what is known about metabolism has gotten to be so complicated that it can't fit on a single chart anymore. There are incredibly elaborate available metabolic models. Those metabolic models, there's one by Roche. Yes, they're and they're so beautiful. I mean, the commercial companies produce beautiful pathways. Often they're nicer to use, nicer than the academic ones to use. So they're great resources. But we're really not in this class going to be focusing on this. People who do very, very large scale metabolic model often do something called flux balance analysis, which gives you steady states and doesn't really think about dynamics much. There are great textbooks on this. You can do it inside of CompuCell, and I have nothing, certainly wouldn't discourage you from doing these things. There tend to be kinds of questions that are a little bit orthogonal to the questions we ask in our systems. But if anybody has ideas about ways to integrate them, we'd be very happy to do that. Fundamentally, metabolic processes in the cell create components, both for building the cell, the structural components of the cell, and also for the energy budget of the cell, in particular ATP and NADH, which are the two classic energy currencies of the cell. They're highly involved with cell cycle, with cell proliferation, cell survival. In the mitochondria, mitochondrial permeabilization is one of the main triggers of cell death when cells are infected or exposed to reactive oxygen species. And so those certainly are considerations that we have to think about. Typically, we're going to be dealing, if at all, with very small components of this enormous set of pathways. But the kinds of questions that we might want to ask are, how does the rate of production of a particular component of interest change depending on the environment? That might be the actual real room temperature, the temperature of the organism. We know that, for example, fever affects the way our body responds to viral and bacterial infections. Substrate availability, in particular glucose and oxygen. We're going to certainly have models in which we consider hypoxia in cells. If we're modeling cancer, we're going to have to consider hypoxia in cells. If we're going to model wound healing and neoangiogenesis, we'll have to consider hypoxia in cells. Availability of glucose is often a limiting factor for cell proliferation. We might want to think about pathway structure. What happens if we add a pathway or knock out a pathway in a particular situation, or use a drug to block a pathway? Again, the complexity here is intimidating, and we're going to normally neglect all of it and assume it works, unless there's a specific small subregion of it that we have to understand for the purposes of our bigger purpose. On the other hand, there are certain basic concepts that we're going to need. They're going to be very, very simple, not those complex models there. And I think it's important that we start out with something There's a comment in the chat, what a friendly image. So I hope what I have here is a more friendly reaction network. I realize that a diagram with one arrow in it doesn't seem like a network, but that's where things start. A network consists of nodes and links. And this diagram has 4 nodes, A, B, C, and D, and it has one link, the arrow. And so we do have to think a little bit about how we're going to describe chemical reactions, because all of our networks are going to be built around the concept of reactions. That is very convenient in some cases. It's a little bit awkward in other cases. People may be used to thinking about ordinary differential equation representations of networks. And I want to say from the beginning that you can always translate a network diagram like this one into an ODE, but you can't always go backwards. And so the network is in fact more fundamental than the mathematical representation. So let's imagine that we have a chemical A and a chemical B. They react to form one molecule of A and one molecule of B, go to form one molecule of C and 2 molecules of D. That's the basic syntax that we're going to use. This is called a rate law. We haven't said anything about what the rate of reaction is. That rate of reaction probably depends on the concentration of A, the concentration of B, the pH, the temperature, the pressure, maybe on light, maybe on whether there's a catalyst present, and so on. But we're just going to now abstract this concept. And to begin with, we have this concept of a node, which is an object, and a link, which is a process. And the links typically here are going to represent chemical reactions, transformations of one thing into another. And what's critical about a reaction is that the thing on the left, on the source side of the arrow, is destroyed, and the thing on the right, the pointed end of the arrow, is created. And you'll see double-headed arrows for bidirectional reactions. For example, 2-ADP goes to one molecule of ATP and one molecule AMP. That makes sense if you think that the ADP is adenosine diphosphate, ATP is adenosine triphosphate, and AMP is adenosine monophosphate. And so the number of phosphates is conserved. It's moving the phosphate around. Or I could have 2 A's go to B reversibly, B goes to 3C reversibly, C plus A goes to D reversibly. Sometimes you'll see multi-headed arrows to represent what's called the stoichiometry of the reaction. The key thing is that any link, any process, has a rate of reaction. And one problem is that people don't always write those rates on the arrows. They should. And they're going to find that in the literature, there's a lot of inconsistency in the notation for arrows. We do have to immediately think about reversible and irreversible reactions. When we put these things into mathematical and computational form, we are going to represent a reversible reaction as 2 separate irreversible reactions, the forward and the backward reaction. Forward, by convention, goes left to right. Backward goes right to left. I guess if we lived in a country where you wrote in the other direction, it would go the other way. But that's where we are here. And so I will try always to write the rate on top of the arrow. If it's a bidirectional reaction, you'll write the reverse rate underneath the arrow. The net rate of reaction is the difference between the forward and the reverse rates. Positive rate means that there's a flow of mass from left to right. Negative means a flow of mass from right to left. There's a term which you'll hear bandied about a lot, fancy term for a simple concept, stoichiometry, which is simply the number of atoms that are used up, or the number of individuals that are used up if you're doing something more abstract, or created on either side. We're assuming that atoms are unitary, and so those numbers are integers, although sometimes chemists like to write fractions, 3.5 molecules of something. You can always multiply up to make those integers. So in this case, 4 molecules of A and 1 molecule of B, 4 atoms of A and 4 atoms, I should be careful. It is molecules here. 4 molecules of A and 4 molecules of B. I was thinking of H2O, so that's why I was thinking atoms. 4 molecules of A and 1 molecule of B produce 2 molecules of A, 3 of B, and 1 of C. That may seem a bit strange. Why wouldn't you net out the production and destruction of A and say that you start out with 2A and wind up with none? And the answer is that the rate laws that you will derive will depend on the total number of molecular species involved in the reaction. And so 2A plus B goes to no A plus 3B is going to have a different rate from 4A plus B goes to 2A plus 3B. So one does have to think about that. What's called the species stoichiometric coefficient is the difference, the net creation or destruction of a particular species. It's relatively common in biology to have a species show up both as a reactant and a product. When you do chemistry in a chemistry course, it tends not to be done that way. But in biology, it's pretty often to have a reaction that includes the same thing on both sides. And so we have to get used to that. If we want to start converting our reaction into something that is computable, we have to think about how to do that. The first fundamental concept is a rate of reaction. Here, we're going to write that rate of reaction on top of the arrow. And typically, again, there are going to be external factors like temperature, pH, pressure that are going to affect the rate of reaction. But typically, that rate of reaction is going to primarily depend on the concentrations of the reactants, not so much usually on the rate, the concentration of the product. If there's a back reaction, that will depend on the concentration of the product. And so we can write A plus 2B goes to 3C at a rate, VF, V forward, that depends on the concentration of A and B. And that one arrow diagram would give rise, if we express this in a continuous form, that's assuming our concentrations are big enough that we can treat the molecular species as continuous as opposed to individual molecules interacting. 3 ODEs, the destruction of A, the destruction of B, and the creation of C. And you'll notice in those rate laws that the dA by dt is minus VF, dB by dt is minus 2 times VF, because I use up 2 molecules of B for each reaction that occurs, and I create 3 of C. And so you have to do a certain amount of bookkeeping when you go from the arrow diagram to the ODEs. And that's one of the reasons that it's difficult to go backwards. We'll find that the software we're going to use will handle all of that automatically for us, which is very convenient. You could have, in principle, a very complex chemical reaction. No, 9 molecules is just pulled out of the air. 9 molecules of A plus 16 of B plus 4 of C plus G go to something else. And if we don't know anything else, we'd say, how do I know what the rate should be? And if there are no intermediates, if that reaction happens in one step, probably doesn't for that complex reaction, but if it did, there is a very simple approximation that works surprisingly well. And there's some textbooks where the entire textbook only uses this approximation, and it's called mass action. And mass action is fundamentally based on the idea of 2 things. One, If I don't have any A or B, the rate of reaction has to be 0. I can't be producing things on the right if I don't have substrate on the left, don't have reactant on the left. And so I need some functional form that goes to 0. 2, if I think about chemical reactions, and there's a bit of, I wouldn't say magic, but a little bit of poorly understood detail here. If I think about a reaction that requires one molecule of A and one molecule of B to happen, then if I think about that as individual molecules, not about concentrations, I need to have the molecule, a molecule of A and a molecule of B come close together. And if I now ask the question, what's the probability of them getting close together? Well, that's going to depend on their typical separation. Now, if I want to have 2 molecules of A, and that separation is going to be inversely proportional to their concentration. If I need 1 molecule of A and 1 molecule of B to be near each other, the probability of that happening is proportional to the product of the concentration of A and B. If I need 2 molecules of A to be near each other, the probability of that happening is proportional to A squared, the concentration of A times A. And so the law of mass action says that the probability of a reaction occurring is proportional to a rate constant, K, some lowercase k, we're going to see lots of Ks over the next day, times the concentration of each species raised to its stoichiometry. So if I need 9 molecules of A, then the rate will go like A to the 9th. Whereas if the concentration of A is low, it doesn't happen. It increases very rapidly as I increase the concentration of A. If I really need 16 molecules of B all to be in the same place, then the rate of reaction goes like B to the 16th, and so on. And so this is called the law of mass action. And for low concentrations of molecules in a simple solution like water, this is very, very well observed experimentally. When I go to high concentrations of molecules, if the size of my molecules is very different, I have a huge molecule and a tiny molecule. If I'm near a surface, if I am in a crowded environment with other molecular species, then there are deviations. Typically, the rate of reaction will be lower than that predicted by mass action. And if the reaction occurs in stages, there are additional considerations. But the law of mass action is a very good place to start. Just the way when you're doing diffusion calculations, you use the Stokes-Einstein approximation for diffusion. It gives you an upper bound on the diffusion rate of a chemical species. Same here with mass action. Typically, the mass action rate will be an upper bound on the rate of reaction. And it's not a bad place to start for chemical reactions. In fact, it's such a common assumption that people often will just write K. And so if you see a chemical reaction like this and they don't write V, but they just write the lowercase k, that means that you're supposed to interpret that arrow as happening with mass action. Actually, determining the values of K is pretty hard. And if I had more time, I'd talk about all of that. If you want any of the standard math bio textbooks, we'll go into the details. So now, if I see an arrow diagram here, A plus 2B goes to 3C, and I say I have mass action, then the rate of reaction is K forward times A times B squared. And again, that will immediately generate the ODEs that I want. If I have a reversible reaction, then I have to remember that mass action does something interesting, which is 2 ADP, the forward rate will be K forward times the concentration of ADP squared. The backward rate will be K backward times the concentration of ATP times the concentration of ANP. And so I do have to remember that the forward and backward rates have different forms. And so this is writing that out with mass action. The forward rates are given by Kf A squared. In this case, I just have said 2 molecules of A produce B plus 2C. The backward rate is Kr B times C squared. I write the forward law for A, B, C, the backward for A, B, C. I have to remember that the forward rate, I consume 2 A's, I produce 1 B and 2 C's. And similarly, for the backward, I produce 2 A's, I consume 1 B, and I consume 2 C's. And I add everything up to get the net rates of reaction. Again, if I look at those totals there as ODEs, in this particular case, I could back out what the arrow diagram was. But in more complex systems, I couldn't do that. Okay. You also have something called catalysis, which is that the rate law might depend on the concentration of a particular molecule that's not used up in the reaction. I'm on a surface. I'm on a fiber. In that case, there's a regulatory arrow written with a lollipop that affects the rate of reaction, here S1 going to S2. But A is not used up in the reaction. And so one of the things that we always have to remember, and people are not always careful about this, is that arrows should always be of 2 types. An arrow with a pointed end on it means that there is transformation or transport of a chemical species. You shouldn't use a pointed arrow to represent regulation. In our world, we always use the lollipop arrow to represent activation, and a T or blunt-headed arrow to represent inhibition. There are a variety of conventions about how to draw metabolic networks. As they say, the great thing about standards is there's so many ones for the same thing. One of them is Systems Biology Graphical Notation Standard, and that is not universally used, but it's not bad. And there are software tools available, some free ones for laying out diagrams of that kind. A concept that we will run into, which I can't go into a lot of detail on today, but is important, is the concept of chemical equilibrium. This is a set of important questions if you're trying to build intuition about what your model should or shouldn't do. And you want to ask the question, is my model reasonable or not? It's important to ask the question, what would happen at long times for chemical reaction? If I start out with chemical species A and B, and it's a closed system, and I let them react, after some time, the reaction will reach equilibrium, where the forward rate and the backward rate are equal. And in this case, I'll get something called K-eq, the equilibrium constant. These are written with capital Ks. And you'll see a lot of capital Ks, which have to do with levels that are reference levels for chemical species, in particular Michaelis or Hill constants as well. And in this case, it's very simple to calculate that at equilibrium, the amount of B I have divided by the amount of A I have is equal to K forward divided by K reverse. This is something that's an easy experiment to do. The problem is it gives us the ratio of the forward and backward rates. It doesn't give them individually. And so equilibrium measurements can give us ratios of rates, but they can't give us the forward and backward rates separately. And that's the main reason that measuring forward and backward rates independently is so difficult. But it does give you a little bit of an idea of K equilibrium. One thing that you'll find if you look at the literature is that these capital Ks all have different units. The lowercase k's are always molecules per time, which is pretty well behaved. But the uppercase Ks have different units depending on the reaction. And so you have to watch out for that. Something that's important biologically is that in biology, the only equilibrium is death, and even that's not really an equilibrium. Biologically, we always have material coming into our system, material going out of our system. And that also can reach a time invariant situation, but it's not one where the system has stopped doing things of interest. It's a balance situation. You'll see these arrows with nothing on one end or the other. An input arrow, like the one on the left here, source K1 going to A, means there's a constant supply of A coming into our system. B going to nothing means that B is either being taken out of the system or being broken down. But in any case, whatever happens at the other end of the arrow isn't of interest to us. And so this could be inflow of energy into our system, inflow of mass into our system, outflow of energy or mass from the system. In this case, we could still reach a steady state, but it's not a chemical equilibrium. One can evaluate again with some simple arithmetic what the equilibrium concentration, I said equilibrium, the steady state concentrations of A and B are. Those are usually written with a little star, superscript star. And even for relatively complex reactions, it's relatively easy to evaluate what those steady state values are. And it's good at practice to learn a little bit about the mathematics of how to do that. Something that we're going to typically neglect, although Julio will talk about this, I hope, a little bit later in the class, is that chemical reactions within cells are often spatially and temporally separated from each other. In particular, the mitochondria are where most of metabolism occurs, and they're separated by multiple membranes from the cytoplasm of the cell. You have the Golgi apparatus, where a lot of post-translational modification occurs, and also transport occurs, that are also separated from the cytoplasm of the cell. You have vesicles for bringing material into cells, for destroying material, for taking material out of cells. We will almost always neglect those, although it's possible to build simulations that do, in fact, represent those processes. When we need to, we should be aware of that spatial segregation. It's remarkable how well network models work, neglecting spatial segregation. But sometimes you'll need to have, for example, a nuclear compartment in your cell, or a mitochondrial compartment in your cell. You may represent that spatially, or it may be a concept of an abstract compartment in your network. But you do have to be aware of that. There could also be gradients of composition within cells. Those gradients can be quite important for things like cell planar polarity or cell motility. Sometimes we'll model them explicitly. A lot of the time we'll abstract them in a variety of ways. And so we need to be aware of the fact that there's a huge degree of structural complexity that we typically neglect. Again, if we think about driving our car, the Baroque architecture of a modern internal combustion engine is something that we typically don't think about. The particular way the mechanics of our engine are structured are things that we tend to abstract away. Even if we think about the engine having fuel brought into it, having the fuel explode, and driving the rotation of the engine, we might not think about the particular way each component of the engine is machined in its shape. And so again, the level of detail we need will depend on the questions we ask. I'd like to talk briefly about the other kinds of networks that we're going to be dealing with. Signaling networks are primarily involved in the flow of information, and we should really think of signaling networks also as connecting to the outside world. Signals, we talked about this earlier, about where does a signal start in the environment, what cells send it, or what material sends it, how is it transmitted in the environment? By advection, by diffusion, by something else, by direct contact. How is it received? Cells have a variety of transmembrane receptor proteins. We'll talk later today about a specific one, a pair of them, delta and notch. Those transmembrane receptor proteins mediate between signals in the outside world and signals inside the cell. They tend to send signals through phosphorylation cascades. The classic example of a phosphorylation cascade is the MAP kinase cascade, where you have the phosphorylation of MAP kinase. Phosphorylated MAP kinase transfers its phosphate to MAP kinase. Phosphorylated MAP kinase transfers its phosphate to MAP kinase, and phosphorylated MAP kinase then does other things. And so you'll see these cascades of signaling, often through the transfer of phosphates, but often through other mechanisms as well. And this is one of the ways that cells send signals within themselves. You could ask, why do you have all of these cascades? One of the things is that they modify the speed of signal transduction. They modify how sharply the final signal depends on the source signal. You want a linear response, that the response is linearly proportional to the extracellular concentration of the molecule, or do you want something that's step-like, where it's an all or none response at some threshold? These kinds of cascades allow for that kind of control. They can be responsible for noise filtering, because these systems have huge fluctuations and they still need to respond reliably. They also allow intervention, so that the rate at which a signal is transduced can depend on a whole host of other factors that would modulate, for example, the concentration of MAP kinase in the system. And so the rate at which the initial signal leads to a final signal could be regulated by the concentration of one of these intermediates. We will do a lot, typically in the models that we build, of protein signaling. Ultimately, we can ask rather simple outcome questions. For example, if we increase the level of growth factor in a cell environment, or toxin in the cell environment, does the cell proliferate more or less? Does the cell die more frequently or less? What happens if we change the density or number of the receptors on the cell surface? The more receptors we have, the more signal we're going to get for a given amount of or level of concentration of a ligand in the environment. You might ask, what happens if we change the number of cell receptors, and how it affects the cell's ability to migrate if we're going to do chemotaxis. If we have a mutation in one component of the system, does it change the binding affinity of the receptor? Does it increase the signal produced by the receptor, or decrease it for a given binding event? We know that in cancer, sometimes mutations lead to, for example, certain growth factor pathways being turned on all the time, rather than responding to whether there is or is not a growth factor in the environment. And more generally, if we perturb this network by adding or subtracting to it, does it change particular cell behaviors in ways that could lead to disease states, or ways we could fix disease states? Notationally, signaling networks typically have the form that we just saw. We have a molecular species S1 that can be transformed into something else, S2. That transformation may be due to reaction to a component, or it may be due to the presence of a component that's not used up. We often have to ask that question. Sometimes we'll neglect that when we think about it. We have a concept of an activator, again, our lollipop arrow, an inhibitor, T arrow or blunt arrow. An activator means that the more A we have, the faster, in this case, S1 turns into S2. There might be some basal rate, so even if there were no A, there'd still be some low rate at which S1 became S2. But we assume that the rate of reaction will increase. Clearly, there has to be a limit. If we had an infinite amount of A, we couldn't have an infinite rate of S1 becoming S2. Sometimes that will be written simply as A promotes S2. That has to be interpreted as chemical reaction. Nothing goes to S2 at a rate depending on A. And that really is just a statement that nothing goes to S2 and that our rate depends on A. In the case of inhibition, there has to be some native rate at which S1 becomes S2. As the rate R goes up, the rate of reaction goes down. If R inhibits S2 and we don't know anything else, we think of nothing going to S2 at a rate that decreases as R increases. Again, since R or A are not used up, R shows up or A show up in the rate law, but they don't show up in the arrow itself. Something that we'll use repeatedly in the course is a way of quantifying our ignorance about rates. There are two forms, which are actually versions of the same fundamental mathematical equation, called a Hill equation or a Michaelis-Menten equation. These actually can be derived in specific cases for specific chemical reactions, although we tend to use them more generically as ways of parameterizing our limited understanding. If we believe that for concentration S of 0, the rate of reaction is 0, and we believe that for large values of S, the rate of reaction is 1, then we can constrain the shape of the reaction curve. And we could say that there are quite a few possible ways of writing a mathematical form, but the simplest one that we could write would be to say that there's some maximum rate of velocity of reaction, Vmax, times concentration of S to the nth power, divided by a capital K to the nth power plus S to the n. And what does the functional form of that do? If S is 1, then I have Vmax times S over K plus S. If S equals K, then that's equal to Vmax over 2. If S is 0, it's 0. If S goes to infinity, then it gives me Vmax. And so this has the form of being 0 at 0, Vmax, or 1, if I take out the Vmax at infinity, and 1/2 of Vmax when S is equal to capital K. If I make that exponent n bigger than 1, it still has that property, but the difference is that if n is equal to 1, the slope, the rate of increase of the rate of reaction as a function of S, is maximal at 0 if n is 1, and it's 0 if n is bigger than 1. And so Hill equations are sigmoids. Michaelis-Menten equations are continuous curves that have maximal slope at 0 and decreasing slope as we go up. And we'll use these Michaelis forms over and over again. The great advantage of Michaelis form or Hillock form is that there only are two parameters involved. K, the value at which my reaction is half of its maximal rate, and n, the steepness of the curve. And so we're able to crystallize our limited understanding into two unknowns. And that makes it a lot easier to fit things. And so we can now, if we assume that that's what's going on, if we see a diagram that says A promotes S, we interpret that as nothing goes to S, promoted by A. We then write that as the rate of production of S is some basal rate, B1, plus Vmax times AN over Kn plus AN. And so now we've gone from an arrow diagram where we knew nothing at all to a mathematical form that has, in this case, 4 unknowns, B1, Vmax, K, capital K, and N. It'll wind up that the value of N is not usually that critical. The steepness is typically not that critical. We often will assume that that basal rate is 0. So we'll make B1 0. In that case, all we have to do is ask the question, what is the concentration of A at which our rate of reaction is half of its maximum? And typically we will scale all of our concentrations between 0 and 1, and we'll make all of our Ks 1/2. And so you can actually begin to build models that make sense, knowing nothing about the system other than where the reactions are. In reality, to make them fit biology, you have to do a lot more than that. But you can build circuits by setting all of your Ks to 1/2, all your Bs to 0, that will make sense and give you a place to perturb around and study around. I'm not going to go into the inhibition. The inhibition has the same structure as the activation. And so in this case, perhaps if you have, maybe the second line is worth looking at. If you have mass action, S1 going to S2 without any regulation would have a rate Vmax times S1. And here I'll multiply by my Hill function, something that I didn't emphasize, which I'll come back to just show you here, is one of the nice things about Hill functions or Michaelis-Menten functions is they have a very symmetric form when I go from activation to inhibition. If I have activating Michaelis or Hill function, which is 0 when the concentration is 0 and 1 at infinity, it has the form a to the n over k to the n plus a to the n. If I want to make an inhibitor, I just replace the a in the numerator by k. The denominator stays the same. And so switching from activator to inhibitor just changes one value, whether I have the variable or the parameter in the numerator. Again, signaling is spatial. There'll be particular domains on our cell membrane that have receptors, other ones that don't. We often will neglect that. Very often, signaling depends on what those receptors are connected to on the cytoskeletal, on the cytoplasmic side, in particular, whether they're connected to the cytoskeleton. Most models are going to neglect that. Occasionally, we'll say that there are patches of the cell that have receptors and others that don't. Gilberto's models of cell crawling, in a sense, are doing exactly that. There's some basic mechanisms that come up again and again in signaling. The formation of complexes, where two molecules bind to each other reversibly. So it's not a chemical reaction, but they form a complex. We'll typically treat that as if it were a chemical reaction. The written notation for chemical reactions is awkward. For complexes is awkward because when you write the complex, it looks like you've written the product of the concentrations. And so people often use a square bracket notation to try to simplify that. We've talked about phosphorylation by kinases. That's probably the single most important thing that happens in signaling. You take there's a kinase that takes a molecule and adds a phosphate group to it. There also are phosphatases that take that phosphate off in an active way. Ubiquitination is a process that tags a molecule with an ubiquitin tail that directs it to be broken down by the cell. And sometimes the phosphorylation of a molecule also tags it for breakdown. One thing that you'll find, which is something that's really hard to deal with, is that many molecular species have multiple sites for phosphorylation. that leads to combinatorial catastrophes when you're trying to describe the molecule. If you have 12 phosphorylation sites, you have possibly 2 to the 12th phosphorylation states. And typically, we're going to assume that N phosphates do the same thing regardless of where they are. That may or may not be true. There's actually a nice language called BioNetGen that was designed to handle those sorts of situations. They show up a lot in metabolism. They're something that are quite challenging to model. Let's talk a little bit about gene regulatory networks. I assume that many people in the room have had some biology, but if not, I'll remind you of the central dogma of biology. It's funny to use the word dogma about science. That DNA is transcribed into RNA, that RNA is edited and translated into protein, and that protein then is post-translationally modified. Those multiple steps happen in different places using different machinery. And in different kinds of organisms, there are some minor variations about this. But it means that from going from a gene in your DNA to an active product, whatever it is, takes many steps. Sometimes the active product is an RNA, in which case there are fewer steps, but nevertheless, if it's got a protein intermediate, there are a lot of steps. And if it's got something like a sugar or it's a lipid or a sugar, there are a bunch of other things that are modified that we don't understand so well. And you also have to transport the protein or whatever your product is to the final location. Each one of those steps can be interfered with in a variety of ways. All of them take some amount of time. There's a certain minimum amount of time for transcription and translation. Transcription by RNA polymerases, translation by ribosomes. And so there actually is a fixed delay. Well, not fixed, but there's a minimal delay. If you turn a gene on, there's about 5 to 20 minutes before you produce any protein at all from that gene. And the OD models that we will build will typically not handle delays very well. They'll always assume that you have some production even at early times. There are a lot of different components involved in all of these processes. We're going to neglect almost all of that. For example, we're typically going to assume that we go directly from an activator acts on a gene, and that activator then produces protein. There's a question about delay differential equations. If one needs to, you can use delay differential equations. None of the standard packages for describing biological networks support delay equations. SPML, antimony, CellML do not support delay equations. You can approximate delays by stacking ODEs together. That's not very pretty, but you can do it. And there's a fundamental reason for this. And the reason for this is that while many delay equations are mathematically well behaved, delay equations are fundamentally infinite dimensional. You write the time difference derivative of A equals X. That's a first order differential equation. You write the time derivative of A is equal to A, some function of the value of A at a time in the past. That's now an infinite dimensional equation. And so you've basically gone from a simple equation to a partial differential equation by writing that. You need to know the history of the molecular species to be able to make that deterministic. We often don't do that. And so any delay equation, delays seem very benign. I do something and then 20 minutes later something else happens. But actually dealing with delays is pretty tricky if you want to do it in a situation that works in all situations. So we tend to try to avoid dealing with explicit delays in our networks. CompuCell itself has no problem dealing with delays. There's very easy to write delays in CompuCell. In our Python, we can put timers into cells and say, when X happens, we set a flag. And then sometime after that, we have the cell die, divide, change its state, and so on. But typically in the network specifications themselves, we're not going to write formal delay equations. We're also typically going to assume that a lot of the components involved in things like synthesis are there. We're not going to worry about the concentration of transfer RNAs, the amino acids that are needed to build the protein, and so on, or that there's a shortage of ribosomes, for example. Although if we wanted to model those things explicitly because we knew they were important, we can do it. Gene regulatory networks fundamentally talk about how genes are turned on and off. There's this notation for gene regulatory networks that tries to make them look like integrated circuits, logic gates. In fact, the world is much, much more complicated than what these show. These arrows really are, in this case, a rather poor caricature of the reality. But to try to draw that reality in full detail is very cumbersome. We will assume that genes are turned on and off with some rate that depends on the concentration of the regulators. In reality, transcription at least tends to be Boolean. A gene is either being transcribed or not, and the rate at which the RNA polymerase walks the gene is typically relatively constant. And so this is a case where Boolean models or stochastic Boolean models are quite useful. And I hope that TJ will teach you how to use MabBoss, which is a tool inside which CompuCell works with for modeling stochastic Boolean networks. Today, we're going to be focusing on continuous rate equations rather than stochastic Boolean ones. But they can be converted into each other if you like. If you like, you should think of the continuum approximation as representing an average rate of transcription over some finite amount of time. Gene regulatory networks typically control long-term behaviors like cell differentiation or major transitions, for example, between phases of cell cycle. You'd like to have some idea of what controls the rate of production, for example, of different species, chemical species, that is, between different cell types. What happens to the birth, death, and transitions of a cell as a function of mutating or knocking in or knocking out genes. What the stable cell types are for a given cell in a given situation. There's a beautiful package called Cell Collective that our friend and collaborator in Tom Hellekar's developed, nice open source package that does Boolean gene regulatory network models. And they've been able to show, for example, that T cells have many more cell types than were understood. There are many more stable cell types. You'd like to know how drugs change these behaviors. If you're designing a chemotherapeutic or you're trying to evaluate toxicity. Really, gene regulatory networks are one of the better developed aspects of network modeling. Again, if you look at these very complex gene regulatory networks, we're typically only going to be focusing on a small subset of those changes during a particular developmental or disease process. We'll assume most of them work. If you know a complete network, they're easy to pull in. Again, one of the problems with gene regulatory networks is that they make it look like everything is happening in terms of promoters and inhibitors of particular genes. But a lot of the turn on and turn off of genes is done not by transcription factors or promoters. It's done by methylation of the DNA, which will turn the gene off pretty permanently, or methylation or acetylation of the histone that the gene's wrapped around. And those aren't actually included in this diagram. Neither are the small interfering RNAs. And so actually a huge amount of what is really important about gene regulation is not shown in diagrams of this kind. So one has to be careful, skeptical maybe is a better word, when you see a diagram of this kind and asking the question, is this really representing the important things that are going on in turning gene on and off? You may simply say that under this condition, the gene goes off or on. That's fine. But don't believe these diagrams. They're a little bit seductive, I think, in terms of making things look easier than they are. Fundamentally, though, we're going to be talking about simple situations in which we have some inducer. That inducer turns on the gene. That gene produces messenger RNA, and that produces messenger RNA is translated into protein. With all those additional steps, typically we are going to neglect editing, transport, post-translation, and simply say that molecule I affects the production of protein P, eliminating all those delays and all of those additional steps. It's somewhat surprising to me that this works, but a lot of the time it actually works quite well. There are a lot of different standards for how these can be described. Again, nodes typically are can be the gene, they can be the regulator, they can be the RNA, they can be the resulting protein, and the links could be doing a variety of things. They could be binding to the RNA. They could be binding to the gene to turn it on or off. They could be the RNA being modified. They could be the post-translation protein being modified. And so there's some awkwardness in the notation that we have to live with, that I'm not going to be able to handle for you today. Just let you be aware of it. There are a whole bunch of things that we're going to neglect. Collective effects, as I mentioned, the critical effect that, for example, one key question that has never really been answered effectively in biology is, what is differentiation? We say a cell differentiates from type A to type B. What does that mean? How does it happen? Well, it's associated with certain genes being turned on and certain genes being turned off. It's associated with the cell remodeling itself in particular ways. It's cytoskeleton changing. But how differentiation occurs and how it's, and typically we'll say transcription factor X, SOX6, induces differentiation. Well, SOX6 will bind to some set of promoters and turn certain genes on and off. We know that that's not capable of inducing a stable change in cell behavior. It's not possible. And so exactly how that transient change in cell behavior induced by a promoter of that kind is translated into stable change of cell behavior is not understood. So there's some big gaps in our fundamental biological understanding. And our modeling will have to just accept those uncertainties. And so I've listed here a whole bunch of uncertainties in the issues of modeling gene regulatory networks. If all you say is gene X is turned on or off, it makes life a lot simpler because a lot of these details get hidden. But you do have to ask the question, is that model sufficiently detailed or not for what we need? Okay, if we want to do interpretation, you'll often see a diagram like this. P1 promotes the production of protein P2 at a minimum that we interpret as P1 binding to a promoter site, turning on the transcription of gene 1, which is then produces an RNA that's translated into P2. That represented as a chemical reaction of nothing goes to P2, regulated by P1, which again is nothing goes to P2 with a rate that depends on P1. When we see a diagram like that, we always have to know that that protein decays. If we don't put in that decay, it's never written. If we don't put in that decay, we'll get something that makes no sense. So we have to assume that P2 goes to nothing. We'll normally assume that that is first order kinetics. The rate of decay is a constant times the concentration of P2. Sometimes there's active destruction of P2, in which case that decay rate could be more complicated. And this diagram got a little bit messed up somehow. It's supposed to be labeled RNA there with A there. In this case, we are putting in the messenger RNA step. That effectively creates a delay in the system. So there was that question by Darshan about delay equations. If you explicitly represent the messenger RNA as a step, that gives you a bit of a delay in your system. And that's fairly well behaved. If you want to, you could add a transport or post-translational modification step too, and that'll come much closer to actually representing the true delays that occur in the turning on and turning off of genes to protein. Again, the messenger RNA itself decays. That's a very important method of regulating protein production. It's often neglected in models, as well as the protein. And so this simple statement, P1 promotes P2, actually turns into 4 reactions and 4 rate laws. Nothing goes to the messenger RNA. The messenger RNA goes to nothing. The messenger RNA goes to the protein. The protein goes to nothing. The rate of production of the protein is regulated by messenger RNA. The rate of the messenger RNA production is rated by the regulator. And so I'm not saying it's trivial to determine these things by yourself. I'm not expecting you to be able to do it after one lecture on it, but it's something you're going to run into when you begin to build models. And so it's not worth it. You shouldn't be intimidated by it. You see P1 promotes P2. You say, what do I do with it? There is a workflow for dealing with this. And so the answer is, it's a little bit like reading English, where the pronunciation doesn't map exactly onto the way the letters are spelled. It's a little bit more like reading Chinese. The radicals have some meaning on their own, but each radical can be pronounced in many ways. Each character can have multiple pronunciations and multiple meanings. And so there is a pattern here, but it's not a one-to-one pattern. We have to infer a lot from what we see written. If we want to do this mathematically, we take our idea from before, we write our lack of understanding in the form of these Hill sigmoidal functions. So we say P promotes P2. We write the production of P2 as a Hill function of P1. The more P, there's a basal rate of transcription. In this case, there almost always is. So B1 is usually not 0. We have a sigmoid, so that as we increase the amount of P1, we increase the amount of the P2 that we get, the rate at which we produce P2. Remember, this is an approximation for something being turned on and off. So really, it would be either on or off at any given instant. We have to say that P2 decays. And so that P1 to P2 arrow then will give us a simple rate equation. Here I've done something which looks a little bit funny, and I'm only putting it in there for the people who are more mathematically sophisticated. in terms of this kind of thing, which is that it's usually a good idea to rescale all of your parameters by the decay rate of the protein. And so I've done that here. So B1 has become C1 times KD2. Makes life much easier when you go forward. Again, if you always scale your concentrations between 0 and 1, you can connect things without worrying about it. And then your Ks control the thresholds. The same thing for inhibition. I'm running out of time, so I'm not going to go through the inhibition in detail. If somebody's interested, I'm happy to meet with you and walk through how to translate these kinds of diagrams into equations that you can act on. Something that we do have to deal with, and Herbert has some good, in one of his other textbooks, in his enzyme kinetic textbooks, has some nice things about it. Very often you will have multiple signals acting as activators or inhibitors, either in signaling or especially in gene regulation. And there's a fundamental problem. There's a simple picture here. You see gene GSC, and it has two activators and inhibitor pointing at it. And you notice they've got arrows there, which they shouldn't in their notation, because the promoters aren't used up. We have no idea a priori how those arrows combine. How does the rate depend on the concentration of those three independent things? You could imagine that we need to have both of our activators for our gene to be transcribed. That would be a diagram like this. Or that either activator could separately turn the gene on. This would be an and, the second one would be an or it could be something more complicated. How does the inhibitor affect the activator? There's no universal way to do this. It depends on the particular gene. However, there are a couple of rules of thumb as guesses to get started. The first one is that activators often combine roughly additively. If it's an and, well, then you have to multiply. If it's an or, you add. And so one linear combination would be to say P1 plus P2 to the n in your Hill function. The other one would be to have the P in the end and the P's act separately. And there are other forms that you can use for ands and ors that Herbert will write in. Inhibitors usually dominate. And so usually whatever activator I have, the signal from the activator is multiplied by an inhibitor as an and. And so in that case, suppose I have P1 and P2 activating, I put those together, and I multiply that rate by the inhibitory Hill function. And that's pretty reliable, not always though, but usually inhibition trumps activation. So that's something to remember when you're building these things together. There are typical motifs that you see in genes. Start out with activation and repression by themselves. Multiple control, which, as I say, is a sort of a rat's nest of poorly understood relations. Again, typically people will assume or for activation and for inhibition. Sometimes they'll assume and also for activation. So you need all the activators and no inhibitors for things to happen. It's very common for a gene to produce a product that activates or inhibits another gene. It's very common for a gene to produce a product that inhibits itself. That's a way of regulating the amount of the product. That's actually pretty accurate. There can be extra factors that come in and affect whether a given molecule actually activates or inhibits. Phosphorylation of the activator or the inhibition is often very common. You phosphorylate the activator to make it work, or phosphorylate the activator to keep it from working. Both of those occur. Again, we're not going to be talking about it today, but if you want to model stochastic Boolean networks for gene regulation, CompuCell does support that. And TJ, I hope, will be talking about that later in the week. The last class, and I'm out of time, but I'll just talk about it very quickly, is physiologically based pharmacokinetic models. These are transport models used dominantly in the pharma industry. If you know PBPK modeling, you can get $150,000 a year job with a pharma company immediately, maybe more, because now the FDA requires PBPK studies for every new drug that's introduced. Again, these model how chemicals are taken up by the body, how they're distributed through the body, how they're metabolized and eliminated by the body. And primarily that's through blood. Some of that's in the urine, the feces, through sweat, through exhalation, through lymph. And they're pretty standardized. We asked, what's the time series concentration of a particular molecule, a particular organ? How does the concentration depend on age, sex, weight, whether you have heart disease? What's the difference between an injectable drug or an orally delivered drug? How long does the drug stay in the body? Does it have any toxic metabolites? Targeting can be a big deal. Chemotherapeutics are usually cytotoxic. They're very poisonous. If you can target them to the cancer and keep them out of the rest of your body, you can use much higher concentrations. And that means they're much more likely to work. And so formulation, binding drugs, either to passive transporters, like particles, or to active transporters that then release them, is a big part of modern biochemistry and modern drug design. Mathematically, transport is pretty straightforward. It's always linear, almost always linear at moderate concentrations. You have what's called fixed law across the boundary. You have a forward rate that's proportional to concentration in one compartment, backward rate proportional to concentration on the other. The big thing about dealing with compartments is that typically you're converting between concentration and amounts a lot. You know how much drug you took in amount of molecule, and you want to know the concentration at the target. And that means you need to know the volumes of different components. And that conversion by volume can get a little finicky. It's just bookkeeping, it's just multiplication and division. But doing the bookkeeping is a little bit tricky. Ultimately, these networks have to work together. Almost all of the problems that we're going to address have some transport, some chemical transformation, some activation and inhibition, some change of gene activity. And so we're almost always going to have to think about very limited, but nevertheless significant aspects of all four of these kinds of processes: chemical transformation, signaling, gene regulation, and transport. So that's what I wanted to say as an introduction to building network models. Again, depending on your background, that may have been blindingly obvious, or it may have gone too fast. I was trying to target people who have some familiarity with these ideas, but may not have put them in practice in the context specifically of building mathematical models of them. I do encourage you to read, depending on your background, a classic textbook like Gilbert Developmental Biology or Molecular Biology of the Cell, and also any one of the numerous really good textbooks on network modeling. Paulson, Uri Alam, Herbert Sauro all have great textbooks. If anybody has a suggestion for a textbook in this area that they really like, either on the biology, the mathematics, or the computing, please feel free to share it in the chat now, or send it to us, and we'll send it around to the rest of the community.